Уважаеми колеги, продължаваме с следващата сесия, която е особено актуална в тези дни на Covid-19 пандемията, която обхвана целия свят. Разбира се, ние не сме пощадени, както виждате при нас случаите нарастват експоненциално по същия начин, както и в цяла Европа и по света. Като ние ще бъдем модератори заедно с професор Стоянов.
Здравейте от мен.
Здравей, Валио. И ще се постараем да дадем някои разяснения за COVID-инфекцията и това какво ни касае нас като оториноларинголози най-вече. Имаме и гости от чужбина. Това е доктор Мубараки. И аз искам да започна с неговата презентация, която заглавието е имунен отговор при COVID-19. Please, Dr. Mubaraki, please start your lecture.
Румене, ако позволиш само да го представя накратко, възможно
Just a minute. He will introduce you.
Sorry, Dr. Puya.
Okay.
I'd like to introduce yourself very shortly, in Bulgarian, of course. Доктор Puya Мубараки е консултант по оториноларингология, хирургия на глава и шия в болницата Губило Голато Тадино в Италия. Дано правилно съм я произнесъл. Президент е на една неправителствена организация Нано Сано, вероятно означава здрав нос, която се занимава специално с проучвания върху диагностиката и лечението на заболяванията на околностите кухини и горните дихателни пътища. С това благодаря. Извинявам се още веднъж, професор Бенчев, и the virtual stage is yours, Dr. Puya.
Good morning, everyone. Good morning to the organizer. Thank you to the organizer. Thank you to my dear colleagues, Dehgani who introduced me to this. And good morning, Professor Valentin Stoyanov. I'm very glad to participate in. So I will go directly to my presentation. I will hope that it will not be long. This is based on some facts in regards to the immune response in COVID-19. It's a quite fascinating topic for me. I've been one of the first to be affected from this disease, and I hope that I did some good for the international community by developing and spreading the word of what was happening in March. So I'm the president of the Association Asasano, and I'm one of the frontline COVID-19 Clinical Care Alliance Associated. I'm basically focusing this subjects today for the immune response in COVID-19, and as you see in the background, this is a video tutor of simulated coronavirus based on a new study on the surface of proteins, and it's based on the spike protein. We will go ahead with this for some information. So first of all, I have no conflict of interest, and I want to go briefly to what was happening in 11 March during the world. We had a 121,000 case confirmed, and basically 10,000 in Italy, so quite a lot. And then what was happening the 12th of March, we had an increasing number, 127,000 confirmed cases and 12,000 in Italy. Most of those cases raised in Italy, and that was the main characteristic of what was happening in Italy. And what's happening now, 21st of November, we have 57 million cases worldwide. That's the definition of pandemic, of what's going on. We will go through this. But I don't forget that 12th of March, I sent this, it was 14th of March, I sent this communication to my colleagues in regards to my situation for smell and taste dysfunction. And what was happening to me was just the previous anticipation of my disease. At that time, no information was so much impactful in the society. I was not be able to have a swab for myself to get a diagnosis. So I said to my colleagues around the world, be careful, be careful, be careful, because we as ENT could be potentially affected. And that's how our communication in the Lancet Infectious Disease that basically changed the guidelines for the WHO. And after that, the WHO recognized smell and taste disorders as one of the main characteristics of this disease. And then in the 1st April, I also, declared what was happening in Italy and provided information in regards to the communication that I have from my friends and colleagues. It was 1st of April, I realized that my colleagues from autopsies were carrying on viral thrombophilic vasculitis. We provided those information and was pretty something gold for the future, because then we realized that might be anticoagulation therapy good choices for those patients. And this will lead to the immune response somehow, yes, and has been recognized from the British Medical Journal few weeks ago. Then after that, my interest in COVID-19 raised once again. I participated in a lot of studies, reviews around the world. In fact, COVID-19 changed in our fields, in our topics, in our thoughts, changed a lot from the composition of the disease and the viruses itself. In here, you will see a latching on the ACE2 receptors in the yellow, allows the coronavirus to enter the human cell. So our previous thought in regards spikes receptor to the ACE2 combination was something. Now it's differently, because we know that the spike protein hinges on 3 different places. So what's really happening in the immune response, it depends on something that is called viral load. And why is it important? Because when we inhale, we know that when we inhale, there are some aerodynamic equivalent diameter aerosol droplets that would stay inside of our noses and paranasal sinus and our oropharynx. And those are particles that are more than 15 micron. And we go for lower respiratory tract, and in here, all the particles that are between 2.5 micron will stay, and then alveoli under 2.5 micron. Why is it important? It's basically because we have to focus attention on viral load and the use of mask. We were one of the first to talk about mask. Even in June, we presenting so much importance of the face mask. It wasn't at that time so much impactful. We said, we as an ENT, we have to make some choice and we have to raise our concern in regard to this. We applied for journals, but we didn't have so much attention until October, where we have the call to action published finally for some information. And in fact, the majority of the journals that rejected us now are facing with the CDC that accept the fact that the community use of cloth mask is mandatory and will have a huge impact on what? On a viral load. The viral load is the amount of viruses that goes inside of our nose and oropharynx, that what we are inhaling, it depends and will change the direction of the immune response. So that's important. So wearing mask is one of such simple measures that will have containing the virus, but as Little Prince said, the essentially is invisible to the eye. This is my daughter. It was on April. So how we define immune response in COVID-19, there's a beautiful picture that I'm taking out from our Paul Marik studies from the FLCC, from the AVMS guidelines. This management protocol was from the critical care that was developed in March. There's some huge similarity in a recent paper that has been published in the New England. However, that was based in March. And as you see from this scheme, there's a viral application first stages, which goes from the incubation to the symptomatic phases. Then we will develop an immune dysregulation. But why is this important? It's important because we have to change our mind in regards to these viruses. We have to focus the attention on the viruses' first stages. When we have a huge amount of viruses, a viral load, our relationship and our immune response will change a lot, because that will impact how our immune response will react through a Th1, a Th2, Th7, through a cytotoxic pathway or immune dysregulation. And we'll go through this. There are some theories in regard to this. We know exactly that there are many of them. This is not new. This is something that has been from decades in here. It looks like when we're talking about immune responses and immune system, everything is new, but it's not. It's not because we already know that when we have a T-naive cell, when a macrophage and dendritic cells will go inside to fight against the viruses, it will go and will produce informations and will interact with the viruses by producing different cells. And those cells will lead to a development of different reaction. As we know, when we have some interaction between the cell, there's a co-kissing, there are, hugs and kisses between the macrophages and these viruses, but then the macrophage will interact with the T-naive cells and will lead to differentiation for T cell that will produce plasma cell, immunoglobulin, or will produce by himself mast cell or inflammations, or directly different T regulatory cells or cytotoxic T cell. And that will lead to a huge differences in how our body will respond to this. And this will also lead to the symptomatology, which is going to be different. We know that some patients, the majority of the patient will have an asymptomatic phases. Some of the patient will be mildly symptomatic, other will be moderately, not so many, but severe and critical cases very low. But this is important, because our immune response will lead to this. And this is also important because for the vaccine implementation, we do need to know which pathway our immune response had to go to produce antibodies, might be, or produce cytotoxic cells, we don't know. We don't know exactly what's the right pathway by now. But what we know is that antibody presence still remain after time, and we will go through this. There are some theories beside this. We know that the immune response will have some information when we go directly through the interaction between macrophage and some other cells and some receptors. That's the important part, because in this we will have the interaction between the macrophage through the spike protein of the virus through a toll-like receptor or through a pattern recognition receptor, and that will lead to a different pathway. As we know, sometimes the PRR, that means the pattern recognition receptor, will lead to somehow to an activation of a Th2 immune response, while the TLR, the toll-like receptor, somehow will lead to a production of a different pathway, like through a CD8 pathway, Th1, that will lead to cytotoxic T cells. So this is the first part. But what about the fact that somehow the interaction between the macrophage and the naive T cells? Naive T cells are those cells that that don't know exactly which path should be undertook. However, in the presence of IL-4, interleukin-4, the pathway that usually goes for this are the development of a Th2 response, that in the presence of interleukin-4, interleukin-5, interleukin-10 will lead to the production of B cells and somehow to antibodies. And at the same time, there's exactly the right production of through the interleukin-4 to B cell and to production of antibodies. There's something strange in the disease. We do know that IgM and IgG that usually are produced after quite a bit of time between each other. In this disease, IgM and IgG will be produced directly, simultaneously, or through different hours. What's the meaning of this? Is that when we are focusing our attention on the first part, to the innate arm that we have, this innate arm, this innate response will produce directly a kind of a protection against the viruses after a few hours, while the production of antibodies will take time. So before producing this, a lot of cytokines and interleukin will be produced. And don't forget the humoral immunity through the production of Th2 cells will lead also to the co-stimulation of natural killer cell, mast cell, eosinophil. We actually, while for the Th1, we do know how does this will work. For the IL-4 and the Th2, we actually still don't know how interleukin-4 is produced and how interact with each other. But we do know that interleukin-4 is mandatory for this. Interacting for the production through the interleukin-12, through the cell-mediated immunity, will lead to the production of Th1. And the funny thing is that Th1 directly co-stimulate macrophage by producing interleukin 1, interleukin 6, interleukin 8, TNF, GM-CSF. Those are so important because the immune response that we will produce in this is a direct contact between cells and the production of the disease and to the, not only to the disease, but the viral debris that will attack directly our cells and destruct our cells. And what kind of cells? We have to remember that the immune response will lead to a stimulation through the SARS-CoV-2 to ACE2 receptors and receptor differently. And those ACE2 receptors are based not only in the oropharynx, nasopharynx, also in lungs, in the kidneys, in the guts, in the blood cells, in the brain. So all these targets will be directly affected through these cytotoxic T cell pathways that will lead to the production of an overwhelming activity against these organs, and that will lead to the destruction of them. What is called cytokine storm is because there's an overwhelming production of interleukin and cytotoxic cells against our organs. So this is not the viruses that's causing itself the problem. The problem is the overwhelming response of our immune system against these viruses and ourselves. So while the first stages of the disease are based on the virus, the second part is the immune response, dysregulation that will lead to the problem. So as previously said, the first part, the innate part, directly reactivated after a few hours, while the production of antibodies are needed after 7 and 10 days. And there are times before that to the production. And that's why some of the patients actually goes under cytokine storm so quickly, and they cannot be restored very quickly. And it's not because we don't have antibodies, it's because the overwhelming reaction of our body against the viruses and against ourselves lead to a dysfunction of organs. And this is why moderate severe cases are going to the ICU, because we cannot manage them properly at home. So proper management at home is mandatory. This is a beautiful representation of something that's been released in the Journal of Allergy, which show exactly what I said previously. But I want you to focus on one thing. Eosinophilic count is so important. We know somehow that lower eosinophilic counts were reported in association with severe cases. And I want you to see this. If we go back to my letter to our colleagues, when I was presenting my situation, I told them in here, keep in mind that my eosinophilic count was tripled. And this might be the fact that I was one of the moderate patient, moderate severe, but I was managed at home by myself. But this is the important stuff. There's fact that lower eosinophilic counts are associated with severe cases. So since I was having a huge amount of eosinophilic counts when I had my diagnosis, that's led me directly to the decision making on where I am at with my symptoms and how could I be managed. That's important. I'm not an internal medicine, but we do know science and we do know medicine. We have to make an interpretation of those data that we have. That's important. So there's a spectrum of disease in community. There are people that go directly through a T cell response, cytotoxic T cell. Some of the other, the production of B cells. Some of the other will have no immune response at all. And at the same time, children are those that majority of the time are free of disease somehow. It's because they have less ACE2 receptor. It's because they have a more natural killer cell. And they do also have a frequent respiratory infection that will lead to a memory of innate R. By going directly to the immune response, I will show you some of our studies that we carried on after after a quite time that I got recovered. I still not recovered very well because I have pneumonia, but my focus now is based on COVID-19 and immune responses and olfactory dysfunction. However, this is a preprint that we have submitted months ago. It was September 12. However, we tried to submit it to the Lancet and New England before it was July, when we had the experience of our cohort of patients that we analyzed after time. It was a comprehensive analysis of recovered COVID-19 patients. At the initial time, there were more than 114 patients, but the problem was that to be able to analyze those patients and routinely performing blood samples to analyze antibody persistence, you need a lot of efforts from the people that were scared at that time to tell them, look, can you come here and we would like to analyze your blood sample to see if you still have immunoglobulin? And we reached this in 30 patients. It was 33 patients, but unfortunately at the end were 30. And we analyzed different data from those patients by dividing and analyzing where they were positive to the disease. And those patients were positive between the period of 18 of March and 30 of March. So all those patients were positive at the disease and developed the disease that were scheduled and were classified in 2-3 main categories based on disease severity. They were mild, moderately severe, and critical. We had one patient that were critical and the other moderate, severe, and mild. We analyzed the date between the first swab that was positive, the second was, that means negativity. At that time in Italian society were recommending two different swabs to recognize the negativity or the disease free of those patients. So we analyzed those data, and then we thought that, well, the period of time between the first swab and the confirmation was the viral shedding. But as we know now, the virus shedding is not represented from the time that from the disease onset until the negative swab. The viruses will be cleared after 2 weeks, 3 weeks maximum. What remains in our self, in our nose, oropharynx, and nasopharynx are the viral debris. Inactivated viral debris, but the RT-PCR don't know that. So we're just picking up viruses. So the virus shedding will be lesser than this. However, at that time, we were thinking about this. So then we scheduled those patients. We asked them to do a first blood sample, second blood sample. And interestingly, what have we seen? We divided the patient in 2 groups, analyzing ELISA methodology for a serum sample and CLIA. Those are 2 different methods. Why we use that? Because we would have been sure that there are no differences between modality. That's important because when we assessing blood samples and immune response and immunoglobulin, there might be changes between techniques. However, it didn't has that much changes between them. And what can you see from here? You will see the impact of the presence and still present of the antibody during the time. And the presence and the persistency of those antibodies not only persist, but also increase. And I remember that when we came out with those information, it was in July, the majority of the paper that were basing their study, they were saying, well, actually, the antibody will drop after 3 months. And I will show you something good that we anticipated at that time. But now, what we do have, how much time does those antibodies will stay up to? And this is fascinating stuff. Actually, we came across this for 7 months, and now we're doing for the 8 months. In regards to the IgM and IgG titers, those right here are my personal one. So those values will represent the ELISA method, the CLIA methodology, in which, as you can clearly see, my IgG is still positive after 8 months. And we are collecting all the data from the same patients that previously were part of our study for consecutive blood sample. This was one of the first report in the month of October. And as you can see, IgG persists during the time. And those are separated in 2 different categories between moderate to severe and mild. So at the end, what I would like to stress you about is that IgG will persist, antibodies will persist more than 8 months. This is the approval that I'm giving to you. This is my personal blood sample, and the data are going to be published soon. And at the same time, this is very important in regards to the thoughts for vax. And why is it important? Do we really think that the development of antibodies are the right pathway, or are the right pathway the Th1, the production of CD8? What we have seen is that the production of antibodies persists in the moderately severe patients, but not in those patients that are mild, that have mild disease. And this is the fact that could be the fact that mild or asymptomatic patients, have a direct contact with the viruses, and our T cell directly fight against them and take care of them. And as we do know, however, a robust T cell response keep going at 7 months. So it doesn't mean that an IgM or antibody production are essential. It might not be the good choices for our immune response. However, we can divide the patient in 2 groups by saying that those patients that have a asymptomatic or a mild disease will produce a direct Th1 pathway, while the moderate to severe will have a different pathway by producing antibodies. How long does those antibodies stay? Well, actually, we are one of the first, and we can tell you that from our studies, our persistency will be more than 8 months. In regards to the fact that how can we address this, we developed a scheme. It's a protocol that I developed with my colleagues from the association. Also, this called Protocol D is based on fundamental as a prophylaxis of early treatment. Early treatment is essential. We've been published also in the national guidelines. I'm taking care of patients from national volleyball team. We treated more than 800 patients. By now, none of those patients went in the ICU or hospital. And that's the fact that mandatory treatment and aggressive treatment are crucial to not developing the disease. And what is important at home is not only the wearing of face masks, but is oximeter. Oximeter will have a huge impact on the decision making. I would love to conclude my presentation because I don't want to be out of my time, just for briefly summarizing the fact that we are providing every 2 weeks webinars. The next 2 presenters will be XIVth Falkens and Joseph Hanna. We're going to talk about EPOS 2020 and Joseph Hanna's implantable devices. Those webinars are free to attend, and you can go to our Facebook pages from Association Azosano to attend. And I would remind you that the previously ERS meeting that were scheduled for Thessaloniki 2020 has been postponed in 2021. And don't forget to attend our ERS meeting in Sofia, Bulgaria for 2023, the 29th Congress of the European Rhinologic Society. Stay safe, and thank you so much for the invitation and the participation.
Well, thank you very much for your presentation. It was quite an experience for us to hear you, to hear about the immune system and its reaction to COVID-19. So thank you very much. Are there any questions from the audience? I don't see any. So thank you again.
Thank you all, and have a nice day. And thank you, Diliana, for inviting me. Thank you so much.
Yes, thank you for coming.
For your excellent lecture. Thank you very much. Thank you.
Thank you.